So we're at the last chapter, chapter 23, the evolution of populations. This chapter might actually help you with this week's lab, hopefully. So one of the most important things to remember about natural selection, that's Darwin's theory about how evolution occurs, is that natural selection acts on individuals, but it is populations that actually evolve. So microevolution, excuse me, is the change in allele frequencies in a population over generations. And there are three mechanisms of change: natural selection, genetic drift, and gene flow. And we're going to be talking about all of those. We know that there is natural genetic variation in any population if you look at the genotypes of individuals. The phenotype of those individuals, their physical appearance, remember, is going to be a combination of their genotype, their genetics, and the environment that they live in. Now, some of the phenotypes that we are going to be talking about and that we have talked about in genetics are from a single gene. You're either this or you're that. Some have two or more genes, and so people exist in a continuum in the population. For example, skin color, intelligence, height. Genetic variation can be measured as gene or nucleotide variability. Gene variability means the average heterozygosity, and this measures the average percentage of loci that are heterozygous in the population. Nucleotide variability is measured by comparing DNA sequences of two or more individuals, but it rarely results in variation in phenotype. And so you might be asking, okay, I don't understand how individuals could have different nucleotide sequences and yet not be that different from a physical standpoint. Well, most of the differences that we see in the nucleotide variability are in the introns, in the non-coding regions of the DNA, so they don't have any effect. Even variations in exons rarely change the amino acid sequence of a protein. If you go back to the protein synthesis sheet and you look at that final page where you can see what messenger RNA codes for what amino acid, you'll notice that there's a lot of similarity in the codons that code for a single amino acid. So you may have the first two letters being the same, with the only difference being that third letter. So it takes a lot of genetic change to actually change the amino acid. The environment can impact phenotype, but does not contribute to evolution. So you got to remember that even though the environment can be the driving force, it still has to, evolution depends on genetically determined variation. Okay. So what are some sources in a population of genetic variation? Well, the first one is, of course, mutation, which is a change in the DNA nucleotide sequence. Then you have gene duplication, where you have copies of the gene that are made. And then you have sexual reproduction. Sexual reproduction evolved specifically to give genetic variation because you are literally recombining existing alleles. So let's talk about mutations for a bit. Mutations are only passed to the offspring if they occur in the cells that are producing gametes. If it is a point mutation, it is a change in only one point, a single nucleotide in the DNA sequence. It can be harmful. It can be hidden in recessive alleles and not seen, but it rarely is a helpful thing. If it occurs in the non-coding regions, we call it a neutral variation because, yes, it is still a variation, but it doesn't give the organism any kind of selective advantage or disadvantage for that matter. Some mutations, remember we just talked about this, are excuse me, are neutral in effect because of the redundancy in the genetic code. Excuse me, the one we just talked about on that sheet where it takes such a drastic change in order to see a change in the amino acids. Chromosomal mutations that delete, disrupt, or rearrange many loci are usually harmful. Duplication of small DNA pieces. Remember, that was the gene duplication. Increases the genome size, but it is less harmful. It may later take on new functions as mutations continue to happen. Mutation rates are very low in plants and animals. Usually, there's one for every 100,000 genes per generation. Mutation rates are even lower in prokaryotes, but they have a short generation time, so this allows accumulations of mutations very quickly. Viruses are interesting, and boy, aren't we experiencing the effect of this. Viruses have very high mutation rates. You know, you're probably already familiar. Even if you get a flu shot, the virus can mutate during the flu season, and you can still get sick from it. And this causes a lot of people to say, well, the flu shot has no value. Well, that's just not true. You're still going to have protection. It just may not be against the entire, you're going to get sick, you just won't get as sick. High mutation rates plus very short generation times, which allows them to accumulate mutations very, very quickly and evade any vaccines that are possible for us to create. You know, this is one reason that we don't have a definitive vaccine for HIV/AIDS, because it continues to mutate at such a rapid rate. Now, sexual reproduction recombines alleles in very unique ways, and that variation comes from crossing over. Now, hopefully you remember that, where that's during meiosis, the formation of gametes, and you actually get the exchange of equivalent pieces of DNA from independent assortment, where genes from mom and dad assort or segregate randomly during anaphase 1 and 2. Fertilization, where you get random egg with random sperm, so each of which are carrying their own unique set of genes. There's a video after that slide that you're probably going to want to watch. Now, this next part is very important and will help you with your lab. Hardy-Weinberg is an equation that we can use to test if a population is evolving. A population is a localized group of individuals, and they're made of one species, and we simply say that that's typically means that they're capable of interbreeding with one another, and very importantly, producing fertile offspring. It's important to remember that a population may or may not be geographically isolated. You know, they may not be separated by the Grand Canyon or a mountain range or be on different islands. Populations still have a greater tendency to breed with members of their own population. Now, your gene pool is all the alleles for all loci in a population. We refer to a locus as being fixed if all the individuals, all of them now, in a population are homozygous for the same allele. If you have two or more alleles for a locus,